All right, so now I don't really have any announcements for once, so we're just going to go ahead and get started. Today what I wanted to do was actually to do a bit of concept mapping based on the terms that you guys began to put together for homework. Okay, so the theme for this particular lecture today are mechanisms of horizontal gene transfer. Let me preface this by saying, as noted in the mini lecture, this is, sorry, less likely to trip if they're over there. This mechanism of genetic information flow is unique to prokaryotes. And there's three major types: transformation, transduction, and conjugation. In a sentence, what is transformation? So how is the information gained by a microorganism in transformation?
It's like picked up from the environment.
Yeah.
Like from lice cells.
Exactly. It's picked up from lice cells. Okay, and this involves some sort of recombination event to occur. In other words, the DNA that's being picked up, there has to be a region of that piece of DNA that is homologous or similar in sequence to the bacterial chromosome to begin with, so that that new piece of DNA it basically gets swapped into the genome and something gets removed. Okay? What is conjugation?
Is that when the cells are linked together?
Yeah, and so we have what we call an F plus or a donor cell, and we have an F minus or a recipient cell. The donor cell has the F plasmid. It also produces the pilus. So this is a structure we talked about a long time ago. The pilus is a hollow protein tube that allows for genetic information to flow from one organism to the next. And in this case, recombination isn't necessarily required. Instead, it's the transfer of a full plasmid from one cell to another, and in the end, both of those cells are considered at that point to be F positive and able to perform conjugation. Okay? And so it's whatever is on that plasmid then is the new genetic information that's being inherited. We can also find on these F plasmids either genes that encode for antibiotic resistance, or we can find genes that are going to contribute to pathogenicity of the organism. So maybe it encodes a toxin, for example. Okay. The last form is transduction. And what is transduction? In transduction, what is actually transferring the genetic information? Ashley? Yeah, a virus, and in particular a bacteriophage, a bacterial virus. And so bacteriophages oftentimes do a really good job at picking up their own genetic information, producing new viral particles. At some low frequency, though, there's a mistake made. And this mistake comes from the fact that one of the things that all bacteriophages do is when they lyse a bacterial cell, they also chop up the DNA into smaller fragments to ensure that that bacterial cell will no longer live. Well, a mistake is made in some cases where instead of incorporating its own genome into the viral particle, instead what happens is part of the bacterial genome gets incorporated. Once that virus infects a new bacterial cell, that it injects that DNA, and again, it requires a recombination with the genome to allow for that new piece of DNA to be incorporated into the genome. What's unique about horizontal gene transfer is, one, it's between species. Or, sorry, I should say not only is it between species, but we also find it within a single generation. A cell doesn't have to go through replication for horizontal gene transfer to occur. Secondly, it doesn't require any sort of mutation either. Instead, what we find with horizontal gene transfer, one of the unique features of this is that microorganisms can gain full new genes that didn't exist in the genome beforehand. And this is another mechanism. These three forms of horizontal gene transfer are all ways in which we think antibiotic resistance actually has spread. Okay? So what I'm going to do is I'm going to just stop here. And what I want you to do is find one other person who worked on the same set of terms as yourself. So if you were working on terms with transformation, find somebody else who worked on terms related to transformation. If you were working on transduction, conjugation, or vertical gene transfer, find one other person. What I want you to do is I'm going to have you take those terms that you put together and create concept maps. We're going to work, I know, we're going to do this for about 15 minutes or so, okay? And then once those are done, we will kind of report out on them. I'm going to take them, I'm going to scan them, they become study tools for later on, okay? And so remember, concept maps, not only the terms, but the connecting idea in between them as well, all right? All right, everybody got partners? If you don't, move around. Don't look at me with such disdain. Anybody do conjugation? Lauren did. Jamie, did you do conjugation? Don't be so angry.
It just made me more confused.
You know what else is hard?
School.
Life. Life is hard. Are you saying bacteria are petty and not life? Here, I'm just those are not ways to organize information.
Yeah. No, like
Then make your concept map organized in this like format. It does, yeah. So there are some cases in which that plasmid will actually redefine into the genome to begin with. In those cases, you do have to have a recombination on the other end. Anybody else have vertical gene transfer? Nobody did vertical gene transfer? Okay. You can go with definite. Okay, go ahead. Transformation. All right, and I'm going to go around and I'm actually going to help answer questions so that this isn't as petty.
As petty.
Y'all be haters. What's that? I will come by and check.
Where did you post the groups for the weekly reviews?
At the end of...
Like, I never know like, what we're doing.
So under the...
What do we put in the center?
There is, all the way at the bottom of it, there's a separate Word document with like the instructions for this and then the which thank you.
Yeah. Every time you look at this page. I was looking I know my storage is full. I had to I know, I wish you could hear it.
Let's go ahead and look at transformation. Let's go to transformation. What terms do we have associated with transformation? let's just start by listing the terms that we've got associated here. Okay, DNA from environment. What else? Competent cell, great term. Others? Guys, I'm not even concept mapping. You don't have to read so much. What else do we have for terms? What requires homology? Others? ATP dependent. Great.
DNA binding protein.
Ooh. DNA binding protein. Recombination. Any others? Potentially between species. Potentially between species, as long as there's homology within the DNA sequence. And this between species is actually true for all forms of this horizontal gene transfer. We can actually get genes swapping between, for example, Staph epidermidis and Staph aureus, or between, in some cases, even between different genus of bacteria. Shigella and E. coli are examples in which we've seen actual horizontal gene transfer occurring. So for example, I'll come back to that one in a minute, but in transformation, I'm going to skip ahead real quick. So in transformation, we have, oftentimes we consider what's happening here is that there is a cell that has undergone lysis or died in some form. The DNA oftentimes in this lysis is broken down into smaller fragments. You might have included nuclease with the terms here because nucleases destroy or chew up the DNA into smaller fragments. And then one of those fragments might be incorporated or brought into a cell through a ATP-dependent mechanism through a transport protein. Nucleases destroy one of the two strands so that only a single strand of DNA is coming in, and then we get homologous recombination. So there has to be homology between the fragment that's coming in with the currently existing genome, and we get a swap out of one for the other. And this can bring in entirely new genes if what's between those two homologous regions is an entirely or a gene that isn't currently in the genome. There are some great examples of this. We'll take a look at one right at the end of class today. But this right here, cells that are able to take up DNA are considered to be competent. So cells that have these DNA transport mechanisms are naturally competent bacterial cells. Again, Streptococcus is a great example of this. We can also engineer bacterial cells to be artificially competent. So for example, one of the ways that we oftentimes work with E. coli in lab is we put plasmids into them to express genes. And to artificially make E. coli competent, we can treat them with salts so that when we snap heat them and then snap freeze them again, the heat allows for the DNA to get into the cells, and when we cool them, their membranes kind of close up. So we increase and then decrease permeability, allowing for that DNA to become self-replicating, a separate plasmid within the cell. Questions on transformation? So what terms would we associate with this one that are unique to this process, but we don't find in any of the other ones? Devin? Competent cell would definitely be a unique one. That might be the only one. I guess actually, picking up from the environment, something that you mentioned, Kaylin, would probably be unique to this as well. Devin. Or Brendan, sorry. You guys are grouped together, so my mistake.
When we were making the map, we included lice cell.
That would be a great example too, because it requires that dead cell, which oftentimes those dead cells might occur through a lysis process that would allow for that DNA to be released. Absolutely. Absolutely. Let's take a look at transduction. Okay. What terms would we associate with the process of transduction? Bacteriophage. What else? Okay, so we've got maybe nuclease that chops up from the bacteriophage that chop up that DNA into small pieces to begin with. And then we might have nucleases, furthermore, that nucleases can prevent that DNA from even being incorporated into the genome in some cases as well. Absolutely. So we've got nucleases involved in a couple of cases here. One, the mechanism by which bacteriophage will incorporate that or bring that DNA into the into their capsid or their viral structure, and then also as a way to prevent potentially recombination from occurring as well. And there's another term that we would associate with this. Requires some sort of recombination into the genome. What else? What other terms do we associate with this process? Yeah, and actually that's one that is true for all forms of horizontal gene transfer. That resistance to an antibiotic can occur. Actually, this is true also for vertical gene transfer, as we took a look at that on Monday, through mutation. What else is unique to transduction? Or what else do we associate with transduction? Just like transformation across species, we can find transduction occurring. Let me give you one example. So one of the first pathogenic strains of E. coli, the one that I refer to all the time as 0157, this was a case that occurred in the early 1990s. It is basically the case that nearly took Jack in the Box, the food company, and destroyed them for years and years and years. It was a horrible outbreak of 0157 H7 E. coli. One of the things that particular strain of E. coli has is it has two toxin genes that contribute to most of the symptoms associated with it. Those toxin genes are actually not unique to E. coli. They originally came from Shigella, a closely related bacterial species. And looking back at how those genes got in, if you look at what's around that sequence for those toxins, it looks like actually it was potentially a transduction event. That the toxins from Shigella somehow got moved into E. coli through a bacteriophage. And so that would be a classical example of a transduction event, where genes from one species have been moved into another one, again leading to a new function of those bacterial cells that didn't previously exist in those cells. The last one is conjugation. Colloquially, or the jargon term is, this is bacterial sex, or the closest thing we get to bacterial sex in the prokaryotic world. And the pilus that is used between those two cells is sometimes called a sex pilus. That pilus, as we mentioned, is hollow. It allows for genetic information to go from one cell to the next. What terms do we associate with the process of conjugation? And this one probably has the most out of all of them, I think. F plasmid, Emily? Okay, let's just start there. So the F plasmid is what gets transferred. What does the F plus and F minus refer to? The donor and the recipient cell. So the donor, the F plus, has the F plasmid, is capable of producing the pilus because that pilus is encoded for somewhere on this F plasmid here. The F minus, the recipient, is what will receive this plasmid. This is just kind of a general schematic of an F plasmid. Obviously, what we can have on here are additional genes, like antibiotic resistance genes. I do want to point out another term that's unique to this one is the ORIT, or the origin of transfer. What we have is a nuclease that cuts one of the two fragments of DNA here, and in this process, what we get is that almost kind of like a spool of yarn, I guess. This chromosome begins to rotate, allowing for one of those two strands of the F plasmid to be moved to the daughter cell, and then both of them undergo replication and recircularize. At the end of this process, what we get are two F positive cells, if this conjugation has fully occurred. Now, that pilus is a pretty weak connection. It breaks fairly easily, and so we can get cases in which this conjugation event isn't complete or may not be successful. Okay. What other terms do we associate with this process? Absolutely. It requires replication of that F plasmid in both of those cells to create the double-stranded plasmid. What else? Meg? Recombination in some cases, right? We'll come back to that in a minute. Abby, did you have another one? Nuclease required to nick one of those two strands. Any others we're missing right now? Transfer of genes, absolutely. Antibiotic resistance. What's that? New genes, absolutely. Completely new genes into those cells. Possibly between closely related species. Or most often this occurs within the same species, but there are cases that have been reported where conjugation can occur between two closely related bacterial species. In the case of this recombination, this is kind of a unique event in which case somehow the F plasmid has integrated itself into the bacterial chromosome of the F plus cell. And once that happens, these F plus cells become known as HFRs, or high frequency recombination cells. In the case of these HFR cells, when they perform conjugation, they begin to not only transfer parts of that F plasmid, but they also begin to transfer some of their own genome to the F minus cells. Now, the genome is somewhere on the order of 5 million base pairs. To transfer all of that genetic information to the recipient cell would take hours, and the pilus does not stay stable enough for that to occur. So oftentimes what we find is that the pilus will remain stable enough for some genetic information to be passed, and at some point it's aborted because that pilus breaks. In those cases, that genome has to, or that portion of the genome from the HFR cell actually has to be recombined into the F minus cell, and that F minus cell remains F minus. This is kind of a unique situation. I'm not as concerned with you knowing about this form of conjugation, but more the classical form here where the F plasmid is self-replicating on its own. We can finish up today in one of two ways. We can, one, do a Venn diagram, just kind of taking these terms and connecting which of these do we associate with vertical gene transfer versus horizontal gene transfer. Or two, I've got a good, better, best question for us to work on. Which would we prefer? And you have to choose one or the option. There's no option 3 of I don't want to do either. Somebody be bold enough to speak up for everybody else? Good, better, best? All right, wonderful. Leaving wasn't an option either.
Where do you want this to look?
You can just put it right here. Thanks, Michelle. So this one is up for you guys as well on Blackboard, but this one comes directly from the review questions. And so the question is, in a classical example of horizontal gene transfer, Frederick Griffith was studying pathogenicity factors in Strep pneumoniae, which is a naturally competent microbe. What does that tell you? If we describe it as being naturally competent, what does that tell you? What process is occurring here? Transformation, good. So Griffith found that there were 2 strains that he observed of Strep pneumoniae. One that he called rough, because when the colonies grew up, they kind of didn't have a shininess to them. They looked rough. And then smooth. Okay? And the rough were non-pathogenic in mice. The smooth were pathogenic. The smooth had capsules. Okay, so this is going back to a structure we've talked about in the past. Well, the rough ones didn't have the gene to make a capsule. So what he ended up doing was taking heat-killed, smooth Strep pneumoniae. These actually, once he heat-killed them, were no longer able to cause disease, which should make sense. They're dead at that point. Once they've been heat-killed, therefore there's no chance for them to replicate. But when he took rough Strep pneumoniae, those that were non-pathogenic to begin with, and he inoculated those and heat-killed smooth Strep pneumoniae at the same time, so a condition that we would assume does not cause infection, what he found was the mice got sick. Not only did they get sick, but when he took those Strep pneumoniae cells out of the mice, he found live colonies again of smooth and rough, when all he should have seen, given that the smooth were dead, was only rough colonies. And so what it came down to was this was occurring by transformation. The question is, basically the question here is, explain how transformation could have led to these results. Okay? We've only got a couple of minutes, so I'm going to give you 2 to think about it real quick, and then we'll just briefly start to outline what the answer would look like. Actually, I rephrased that. I'm going to give you 3 minutes to work on this, and we'll talk about it first thing on Friday. I want to make sure we have time to cover this, okay? So take 3 minutes to write down some thoughts here, and then we'll finish up on Friday.
Do you want her?
No, that's okay. We covered what we needed to. I'll never do a concept map ever again. Fingers crossed.